Good evening and welcome to Pacific Science Center's virtual Science in the City. I'm Zita Strickland and I am pleased to be your host for tonight's virtual Science in the City talk. Science in the City is made possible with the generous support from Jeff and Carmen Barr. Additional support comes from more than 1,500 individuals, companies, and foundations that donate to Pacific Science Center each year. Tonight's Science in the City will be recorded for later viewing on our YouTube page. And on that page, you can find many of our other past Science in the City talks. Learn about pizza making, robots, Martian landers, and a little closer to home, the Science of Climate Pledge Arena. All these past talks can be found on our Science in the City playlist on the YouTube page for PacSci. In addition to these events, our web pages also contain hands-on activities that can be done at home or in the classroom, and information on our live and interactive virtual field trips. You can learn more about all these programs at pacsci.org. I want to thank everybody for tuning in with us tonight and for being curious with us. Thank you also to all of you who made a donation at the time of registration. If you hadn't yet and would like to donate, you can do so at pacsci.org/support. Your donation helps to ensure that curiosity never closes. Now, tonight's event is kind of becoming an annual tradition as we dive into spooky science, both real and fictitious. Tonight's exploration is a forensic report, and the best investigations are interactive, and we have a couple of methods of interactivity planned for you tonight. First off, throughout tonight's event, you can share your questions to our speaker by adding them directly to the YouTube chat box on your screen. If you don't see the chat box or you can't add to it, it just takes a moment to make a free YouTube account, and then you can type in and leave your question for our speaker to answer during tonight's event. Secondly, during the program, there's going to be a couple of polls and some other interactive features to access digitally. These are going to come to you in two ways: via a QR code on the screen that you can scan with your smartphone, and via a link that we'll put in the YouTube chat. We've got a sample poll for you now to ensure that it's all working smoothly. So we're going to put a link in the chat, and we're also going to pull up a QR code on the screen. So to access that QR code, all you need to do is grab your smartphone, open up the camera app, and point it directly at the QR code that should be loading any moment now. See, and this is why we test. We're also going to see a link in the chat for you to access. So that link is in the chat and you can click on that, and it's a simple form with two questions for you. We also should, maybe we might try again, see if we can get that QR code. Talk about spooky Halloween. I don't know about all of you, but every technology today for me has been full of gremlins. It is Halloween theme for sure. And the gremlins are in the technology. All right, so click on that link, go to the poll with our two simple questions. Question one, we want to know a little bit about who you're watching with tonight. So let us know if you're watching with one person, just yourself, or if there's two of you watching, if there's 3, 4, 5 of you, if there's more than 6 of you. And we also would love to hear what topics you are interested in for future Science in the City programs. You can let us know if there's a Halloween topic you're interested in for next year. We're actually already planning and have some ideas. And there's future Science in the City programs, could be on any topic. We've got our QR code up now. So again, if you prefer to access via the QR code, all you need to do, grab your smartphone, open up the camera app, point it right at the screen as though you're going to take a picture. You should see a little button that pops up that says QR code is detected. You can click on that and that'll take you to the form. Let's see, it looks like quite a few people watching just yourselves. You're not alone though, because we're all watching together. We've got about a third of people are watching with one other person. We've got a few people that are watching in groups of 3 or more. Some great topics. Oh, Kaitlyn, we've got a suggestion for Frankenstein and ghosts next year already. Sea mammals, surprise me. That's my favorite answer. I like that one. We'll see what we can come up with. Go ahead, keep filling that out and keep your smartphones handy and keep that open tab in your browser open because there are going to be several more interactions tonight via QR code. And also remember, you can add questions and comments at any time to the YouTube chat. We're sharing those with our speaker. Now, tonight, I'm going to start out by setting a little context. Remember, this program will be recorded. You could be watching this at a future date beyond today. But today, right now, it's October 26th, 2021. It's 7 p.m. here in Seattle. It's dark outside, and it's actually pretty classic spooky conditions as I look out my window. It's dark, the wind is blowing, leaves are being blown off trees, it's raining off and on. It's the perfect night to investigate the crime of the century, Forensic Report, 1886, the death of Dr. Henry Jekyll. Our guide through this forensic case tonight is Dr. Kaitlyn Casimo, PhD. Kaitlyn leads the training and education at the Allen Institute, where she develops programs, materials, and other resources to bring a variety of open science resources to labs and classrooms around the world. She received her PhD in neuroscience from the University of Washington and studied neuroscience and theater at Pomona College. And when she's not going to the theater, she plays tennis. She volunteers here at Pacific Science Center as a science communication fellow, and she's on the advisory council at Seattle Rep. This is Kaitlyn's, I think we decided it was your 6th Halloween talk with us tonight. Kaitlyn, I'm so excited that you are here with us, and I'm excited to dive into this forensic report.
Thank you. I'm going to share my screen, so hopefully you all can see that now. Yes?
Yes, it looks great.
Great. So thank you everyone for joining me for tonight's forensic report review for the neuroscience sections in case 1886, the death of Dr. Henry Jekyll. I'm Dr. Kaitlyn Casimo. So first, a review of the events. All right, Dr. Henry Jekyll, a notable physician, gained infamy after lurid details emerged that linked him to the notorious murderer, Edward Hyde. Initially, all we knew was that there was a suspicious connection between the respectable professional and the known criminal. Hyde started out by making a nuisance of himself, then escalating to reckless assault, trampling a child, and bribing the family with a check in Jekyll's name, and then escalating to greater violence, and eventually to the apparently random murder of Member of Parliament Danvers Carew. The known connection between Hyde and Jekyll was bad enough, but then a cane belonging to Jekyll was found at the murder scene. Meanwhile, Jekyll had fallen into ill health and withdrawn into seclusion. Between the changes in his behavior and Hyde's increasing violence, Jekyll's friends grew concerned, particularly fellow Dr. Lanyon and his lawyer and friend Utterson, from whom we know most of this information. After Carew's death, Jekyll received a note, supposedly from Hyde, claiming that he would bother Jekyll no longer, after which Jekyll resumed his normal life, but only for a short time. Not long after, he retreated farther into seclusion than ever before. Finally, the details became clear in the accounts of Jekyll's friend Lanyon and of Jekyll himself. Lanyon's account revealed that by some trick of pharmacology, Jekyll had developed a drug that would transform him into Hyde and then back into himself. This transformation affected both his appearance and his behavior, but he kept his memory across the two personas. Lanyon died shortly after finding this out of the great shock, which frankly is pretty understandable. Not long after that, Hyde was found dead in Jekyll's lab, wearing Jekyll's clothes, apparently by suicide, with a letter in Jekyll's handwriting found on the desk. In that letter, Jekyll claimed that he wanted to create a drug that would separate his industrious and upstanding self from his more selfish, base, and by his own description, evil impulses. He rationalized that this would be good for both of his selves. The upstanding self would be free of these unwanted, nasty impulses and consider himself purified. And the ne'er-do-well would be free of the guilt of not doing anything useful and would also not share his identity with the good doctor. This did not go as planned, obviously, with the evil self slowly taking over completely and becoming more evil than Jekyll expected. And Jekyll ultimately lost control of Hyde, first requiring more and more of this drug to transform back into Jekyll, and eventually transforming spontaneously into Hyde with no drug at all. When a new batch of the drug failed to work entirely, fearful of becoming Hyde permanently and what violence he would commit, Jekyll was able to take control while in the form of Hyde and commit suicide, leading to the discovery of Hyde's body. The total dead in this sordid affair, depending on how you count it, was 3 or 4, Lanyon, Carew, and Jekyll/Hyde. This case has made so many headlines that you probably already knew what the early investigators did not, which is that Jekyll was also Hyde. But there's so many open questions in this case that the investigation was ordered and I was brought in, not least of which that we don't have any samples of this drug. And the only people who witnessed the supposed transformation of Jekyll into Hyde, Lanyon, who died of the shock of it, and Jekyll/Hyde himself, are dead. Today, I'm presenting the neuroscience portion of the forensic report, where I will attempt to reconstruct the pharmacology and neuropsychology of these events. I'll also touch on some of the ethical standards and requirements that could have prevented this. For other details of the case besides this neuroscience forensic report, please see the original investigative case by my colleague, Detective R.L. Stevenson, the letters and statements in the case provided by Dr. Jekyll's lawyer, John Utterson, and any other source on the case besides the tabloid reports on this sensationalized events, please. So in today's review, I'm going to present my conclusion on these 3 open questions left by Jekyll's death and with him, the death of Mr. Hyde. 1, what was in that drug that transformed Jekyll into Hyde? 2, what does Hyde's behavior tell us about his mental state and also about what Dr. Jekyll's drug did? And 3, how could Jekyll have been stopped by proper research ethics and safety measures? So first, Dr. Jekyll. We'll start by investigating what Dr. Jekyll called his serum. What was his goal in creating this drug? What were its effects? And can we make a forensic determination of what was in it based on how it acted on the brain? Dr. Jekyll explained his research plan in a letter found after his death. In an effort to separate his upstanding ignoble selves, he developed this drug that would transform him into a different person. So he could go drink and gamble and apparently murder without detection or guilt about not being productive. The violent and physical Hyde behaved very differently from the reserved and intellectual Jekyll, and even had a different physical appearance. We also know that Jekyll needed to inject this drug in order to transform into Hyde, and also gave himself an injection to turn back into Jekyll. We have very little information about his drug discovery and testing process before trying it on himself. So in this section, I will ask, what is in this drug? How did he develop it? And how does the drug act in the brain to achieve the observed effects? First, what is a drug? A drug is any molecule that affects the body by binding or latching onto a free-floating molecule or a cell in your body. This binding process produces the drug's effect. It might increase or decrease the rate of some process in your body, help replace the effects of something that's missing, and many other effects. In this case, the drug seems to have made Hyde, but it seems to have made Hyde reckless, violent, and greedy. That is, very motivated by rewards. These clear behavioral effects mean that the drug is acting on the brain. Any drugs that affect behavior or cognition are called psychotropic drugs. Any molecule that has any effect on the body is considered a drug. It doesn't have to be medicinal with the intent to treat or prevent a disease. It can be an everyday molecule like caffeine. And of course, drugs of abuse also count. But for a drug to have an effect on the brain, first, it has to get there. Getting a drug into the brain is particularly challenging. Drugs have to fit specific criteria in order to actually enter the brain and affect it. Most modes of administering drugs, and this goes for all drugs, not just drugs that affect the brain, lead first to the bloodstream. One of the few things that we do know about Dr. Jekyll's drug is that it was injected intravenously, directly into a vein. That by definition means that it goes directly to the bloodstream. In contrast, drugs delivered via the digestive system, either end, get to the bloodstream via the liver, which can metabolize quite a bit of the drug and either turn it into a byproduct or filter it out entirely before it ever reaches your blood. This is called first path metabolism. Even for all of the other pathways, your blood is filtered via the liver. and this is second pass metabolism. Even once the drug has reached your blood by one of these many methods, it doesn't necessarily affect the brain. Most drugs don't affect the brain. In order to enter the brain, molecules generally have to pass through the blood-brain barrier. Your brain is full of blood vessels carrying blood, oxygen, nutrients, and yes, drugs throughout the brain. Around each blood vessel, the blood-brain barrier is physically formed by the cells of the blood vessel sticking very closely together to each other, plus glia, called astrocytes, that physically surround the blood vessels of the brain and only allow selected molecules through. There's a small list of molecules that your brain wants to have pass through: amino acids, nucleosides, other materials that your brain needs in order to function. They're brought through the blood-brain barrier by transporter molecules that escort approved other molecules through and into the brain. About 98% of all small molecules don't pass through this barrier, and about 100% of larger molecules. Drugs that have an effect on the brain therefore have to be able to sneak through. Most psychotropic drugs take advantage of those transporters meant for desired molecules. If the drug can also interact with a transporter that's meant for something else, it can get passed right through. So, in addition to designing a drug molecule that will have the desired effect in the brain, once it gets there, first it has to be able to get there. And this is a real challenge for designing drugs that are meant to be able to affect the brain. If they can't get in, they can't work. Jekyll got very lucky that his drug was able to cross the blood-brain barrier. For example, dopamine can't cross the blood-brain barrier, but levodopa, also called L-dopa, a precursor molecule that is used in the brain to make dopamine, can. It's used to treat Parkinson's disease, which is caused by dopamine-producing neurons in the substantia nigra dying and leading to a disruption of the basal ganglia circuit. L-dopa is administered orally. So first it has to survive first-pass metabolism, then get through the blood-brain barrier, and then get turned into dopamine. And only about 1% of all L-dopa actually gets to that last stage. Remember L-dopa. I'm going to come back to it later. If you want to bypass the blood-brain barrier, the most practical option is intranasal administration, a spray or powder that's inhaled and absorbed in the mucous membranes of the nose rather than traveling down the airway into the lung like an inhaler. The drug then travels directly to the brain via the olfactory and trigeminal nerves, which are cranial nerves. Cranial nerves connect directly to your brainstem rather than to the spinal cord. They therefore bypass the blood-brain barrier that separates the brain from the rest of the body. Peripheral nerves also encounter the blood-spinal cord barrier. A big limitation of the method of intranasal administration, though, is that the nose is just not that large. So the doses have to be pretty small. And Jekyll's drug, he reported other effects on his body besides his brain. So he wanted that systemic effect he could get from an intravenous route. Different routes of administration of different drugs also lead to different absorption rates and different effects. So, for example, a prescribed opioid designed for home use generally comes in pill form, but may be administered intravenously in the hospital. To reach the same level of pain-killing effect, oral doses are much larger than intravenous doses, on the order of 3 to 4 times larger. Or an intravenous dose of the same size may be considered something like 3 times stronger. Intranasal doses can be even smaller than IV doses to achieve the same effect on the brain. So somehow, Dr. Jekyll's drug was able to cross the blood-brain barrier. He himself had speculated that his drug's effect may have been due to using a tainted ingredient, and perhaps whatever that contaminant was either helped the main ingredient cross the blood-brain barrier, or perhaps accidentally was the active ingredient and the rest of the concoction was doing nothing, which is why when he switched batches to a different ingredient, it didn't work anymore. But the initial batch was in there, it got to his brain, so how does it have its effect? Most drugs in the brain work by binding to transmembrane proteins, neurotransmitter receptors, or reuptake transporters. I'm going to focus on the drugs that work this way because I believe that's how Dr. Jekyll's drug worked. Your brain has somewhere in the neighborhood of 86 billion neurons, each of which can form hundreds to thousands of synapses with other neurons. That's what you see in this image on the left side of the screen. That's one synapse. So focus on that image on the left for now, please. The signals between neurons come in the form of neurotransmitters, which are floating through that little gap there, that create an effect on the receiving cell. The neurotransmitters are manufactured in that neuron from precursor parts like levodopa. The neurotransmitters themselves don't actually pass through the blood-brain barrier. The presynaptic cell, which is the top part of this image, releases a neurotransmitter. And my example cell is using dopamine for a reason, hint, hint. It floats across the synapse and binds to receptors on the postsynaptic or receiving cell, inducing a response. Once that second cell, that postsynaptic cell, has responded and the dopamine's job here is done, it's recycled into the presynaptic cell to be used again by a reuptake transporter. Generally, the signal in a synapse is one directional from pre to postsynaptic. Here in a synapse, a drug can work by binding to a receptor or a reuptake transporter. For example, dopamine reuptake inhibitors will bind to the reuptake transporter in charge of recycling dopamine and prevent it from working, which means that the dopamine is stuck in this synapse and it hangs around for longer, and it potentially activates more receptors on that postsynaptic cell before that recycling happens. There's a few classes of drug that do this, that reduce dopamine reuptake. Some modern antidepressants, though they're not nearly as common as the selective serotonin reuptake inhibitors, SSRIs. Monoamine reuptake inhibitors, which are older antidepressants, they're not as commonly used anymore. And many drugs of abuse, such as cocaine. So now imagine this effect of the dopamine reuptake transporter inhibitor, or something that binds to the dopamine receptor itself, affecting billions of synapses at a rate of 300 times per second or more that this signal happens. Because a drug works by binding to a particular target, you might have already realized that this means that the drug effect is determined by what it binds to. While the effects of a drug might be quite precise in terms of what receptors or reuptake transporters it binds to, the drug doesn't target only specific areas of the brain. The drug's behavioral and cognitive effects depend on its receptor target distribution and not the drug's physical distribution. It doesn't get targeted. So the image on the right side here shows the distribution through the brain of the RNA for D1, which is the most common dopamine receptor. Red and pink means there's more of this receptor, and blue, light blue, means that there's less of it. So you can see that this receptor is quite widespread in the cortex. That's the upper part of the picture. You've got on the left, the outside side of a brain, and on the right, a brain sliced in half down the middle, and you're looking at the inside. But this receptor is most highly concentrated in the basal ganglia and surrounding subcortical regions. That's the bottom left picture. And it's quite rare in the thalamus, brainstem, and cerebellum, which is in the bottom right picture that's all blue. Therefore, a drug that targets D1 is going to have its greatest effect in the brightest red region, which is called the striatum, and some effect in the rest of these less bright red and the pink regions, and pretty minimal to no effect in the blue regions. The behavioral outcomes of this drug then depend on what these regions do. There's a lot of moving targets on psychotropic drugs, drugs that affect the brain and therefore the mind and behavior. So they are particularly difficult to develop. More than a few psychotropic drugs were actually discovered by accident when trying to treat something else, which was sort of the case here. Iproniazid, which was an early antidepressant, was originally being tested for tuberculosis. It didn't do anything for the TB, but it improved patients' mood. And it was also derived from rocket fuel. Chlorpromazine, now used to treat schizophrenia and bipolar, was originally being developed as an antihistamine for allergies. And LSD was supposed to prevent hemorrhages. So we don't actually know what Dr. Jekylls did on a cellular or molecular level, and we don't have any samples of this drug left to test it out. He used the last of it up. But I have my theories, which we'll come back to in the next section and which I've foreshadowed a little bit here. But first, if he had tested the drug properly, what would he have done? Drug development begins with basic research, also called foundational research. This is research that's just exploring how the body works. What do different molecules do? How do organs function? How do body signals interact? What do different parts of the brain do? This process can take a long time, and it may not lead to specific clinical applications like a drug being discovered. Sometimes the purpose is just to understand the world we live in and our bodies a little better. Basic research may occur in cells in a dish or tissue samples, animals, or even humans. I actually spent my research career doing basic research in humans before I got into doing forensic reports like this one. Once we understand body systems, we can develop molecules, drugs that will target specific elements of the body. So now we have an idea of how the body works. We have a molecule that we think will interact with something in the body in a useful way. Next up would be preclinical research on a drug. Does it actually have the effects in cells and bodies that we think it and hope it will? This generally involves animal tests. Sometimes we may trust a drug in multiple species, since different species resemble or don't resemble humans in different ways. For example, while mice are one of the most common research animals, hearing research is often done in guinea pigs because their ears are much more similar to ours. Dr. Jekyll, however, does not appear to have done any basic research or any preclinical research and jumped straight to testing in humans. One human, himself. Clinical trials should consist of 3 phases, which you're probably more familiar with now than you were 2 years ago. In phase 1, the drug is tested in a very small number of healthy people to check for side effects and safety. Anything that gets to a phase 1 trial, we think is safe, but there's enough differences between animals being used in the preclinical research and in humans that we have to check, especially before trying it on a human whose body may be weakened due to an illness. In phase 2, we then work out the dosages. Not so high that it's dangerous, but not so low that it doesn't do anything. This is called the therapeutic window. For example, warfarin, a drug used to treat blood clots, has a famously narrow therapeutic window. Too low a dose and it doesn't do anything. Too high and it can cause excessive bleeding, including bleeding in the brain. Those are hemorrhages and they are bad for you. And finally, in phase 3, we determine whether the drug is actually effective. It also has to be found more effective or have less side effects for the same effectiveness than anything else that's already available. And then if it measures up, it goes through the paperwork and the regulatory process of approval, and it's released. And after it's released, it's monitored for problems. A famous example of a drug that was pulled for problems that came up in this post-surveillance testing was Vioxx, which was for arthritis. And it was linked to an increased risk of heart attacks and stroke. And they discovered this link by doing that post-release surveillance on people taking it. A drug that is not an example of this is thalidomide, which was intended as a sedative and was used off-label, meaning it wasn't tested for that use for morning sickness. And it was never approved in the United States because of a lack of safety data from clinical trials. This testing process and regulatory approval applies to all medications, pills, injections, vaccines, and so forth in the United States, and it's very similar in Dr. Jekyll's home country of the United Kingdom and in many other countries. This process is estimated to cost an average of $1.3 billion for a single drug to go through R&D to wide release in the United States. And that $1.3 billion does not include the basic research done at universities. Only costs incurred by the drug company. And even within a single company, it's difficult to link basic research costs to release drugs, let alone the accounting on the importance of publicly funded basic research. As I mentioned, the primary goal of preclinical and later clinical studies is to determine what the drug does in a whole organism and make sure that it's safe. If he had done any preclinical testing, for example, in mice, Jekyll might also have noticed his test mice going after other mice. He might have also noticed that the doses needed to transform the Jekyll mice into Hyde mice got smaller and smaller over time, while the doses needed to go back from Hyde mouse to Jekyll mouse got larger and larger until his test subjects started turning into Hyde mice without any drug at all and wouldn't turn back as the mice developed tolerance. Obviously, the drug did work in humans, but without testing, he went into this without knowing a safety profile or a mechanism of action or information on dosing. And he didn't understand the cognitive effects. So let's get your opinion. Is this a terrible research plan? So go ahead and open your camera app, point it at the code, and it will take you to a survey, or look in the YouTube chat for the link. This survey is anonymous and completely optional, and we will look at your collective results during the Q&A. So Dr. Jekyll's research plan, as best as we can reconstruct, was step 1, create a drug based on some kind of speculation about the known pharmacology of its ingredients, but we don't know what because he didn't take any notes. Step 2, inject it into himself. Dr. Jekyll apparently did no preclinical basic research, no preclinical research on the drug in animals, no clinical trials, and just went straight to opening his own vein. Post-release surveillance, or post-use in one-person personal letters, reveals at least one serious side effect, which would be transforming into a murderer. So here are some of these questions that you should think about when evaluating this research plan. Was his research question, can he separate the good and evil portions of himself, meaningful? And is it novel? Will this experiment answer his research question? Is the answer going to be robust? Is the experiment safe? And is it being conducted ethically? So you've got a few minutes as we go along to answer that survey question. We will go through the answers at the end. Dr. Jekyll's research process aside, what was in that drug? That was the question I was asking in this section of my report. We don't know exactly, and we never will, because Dr. Jekyll left us no notes, conducted no tests, used up all of his samples, as he describes in his last letter. The safety profile of this drug outside of himself is also not known because he did no animal or clinical tests. We know it must have been a small molecule drug that could cross the blood-brain barrier, but it didn't have to survive first-pass metabolism. Also, no drug known to science can transform a person physically as dramatically and quickly as Jekyll claimed he did when he turned into Hyde. Frankly, I suspect it was all in his behavior, posture, and mannerisms, with the added effect of Hyde's blatantly wicked nature that everyone he met immediately fixated on. So this isn't very much to go on. It's mostly a list of his failures. My hypothesis of the drug's mode of action is exclusively based on its behavioral effects and not on its chemical properties. Unfortunately, that only gives us a general class of drug and not a single specific drug. But I think this drug was a dopamine antagonist. Dopamine, which we looked at in that synapse before, underpins the reward and motor systems of the brain. So what is a reward? What is a reward in terms of your brain? And what happens if we disrupt the reward system as I think has happened here? And why do I think this drug is a dopamine antagonist? So part 2, Mr. Hyde. Jekyll's own stated motive for developing this drug, again, was to separate his upstanding and productive self from his greedy self interested only in his vices. That is, to seek out rewards without consequences. The consequences part didn't work out as planned, but as Hyde, his behavior was certainly reward-oriented. Jekyll and Hyde had separate personalities and physical appearances, but they shared their memory, so Hyde's indulgence satisfied Jekyll's desires too. Hyde's behavior was highly reward-seeking, to the degree of probable dysfunction in the reward system, driving excessive reward-seeking behavior. Though Jekyll never described the details of Hyde's behavior, we know he drank heavily, got pleasure from violence, and who knows what else. I'm going to leave the criminal speculation to the other contributors of this forensic report. He also demonstrated dysfunction in related cognitive functions, particularly impulse control and risk assessment. This is seen in his unconstrained and impulsive violence, trampling children, quick turns to threats and bribes in the face of consequences, and the murder of Danvers Carew by beating him to death in the street and leaving behind easily identifiable evidence. So let's get into Hyde's behavior and what it tells us about his brain state. What is a reward? How is it represented in the brain? How does that link to impulse control? And why do I think it's a dopamine antagonist? What is a reward? A reward is something that your brain responds to positively and wants again. Wanting is a brain state where you're motivated to pursue a reward. And liking is the pleasurable reaction to getting it, a reaction that you want to have happen again. Dopamine system of the brain responds to both wanting, anticipating a reward, and also to getting it, or to getting an unexpectedly large or small reward. The reward itself can be many different things. Concrete rewards include food and water, money, prizes, stuff. More abstract rewards include praise, awards, compliments, exercise, and winning a game. Addictive drugs bypass the normal reward processes and directly increase the amount of dopamine hanging out in your synapses. They stimulate the reward circuits at a very high rate, constantly, to the point where nothing else feels rewarding enough anymore. Reward-seeking behavior can lead to repeating behaviors in order to get a reward again. This is how training works, like for pets. You associate the behavior with the reward, and eventually the rewarded behavior becomes habitual. Reward seeking can also lead to innovative behavior if repeating the original behavior didn't work to get that reward again. Dr. Jekyll kept returning to the drug that turned him into Mr. Hyde. Hyde engaged in rewarding behavior to the point of recklessness, which Dr. Jekyll presumably found rewarding enough too to keep returning to this drug, to return to the Hyde reward-seeking state. So let's review the reward pathway. This image shows most of the major players in the reward pathway. This is a brain sliced in half down the middle, and we are looking at the inside. The reward-related functions in these areas all run on dopamine. There's a lot on this slide, but this is a core pathway of how the brain processes reward, so it is important. Please bear with me. The dopamine system processes rewards, anticipating them, receiving and responding to them, responding to unexpectedly large or small rewards, prioritizing behavior that will lead to them, and associating rewards with emotions and memories. The circuits in this pathway actually respond more strongly to the expectation of a reward than they do to getting the reward. This motivation to pursue is a strong part of this pathway's function. There will be a spike in the reward signal for an unexpectedly large reward and a drop for a too small one as well. Dopamine is also involved in movement, which makes sense as pursuing a reward is going to require movement. Several of these reward pathways are pathway areas are part of the basal ganglia, a group of regions principally known for their role in refining motor commands and making them more precise, among other functions, and increasingly of interest for their role in rewards. The reward pathway also works closely with the limbic system regions responsible for emotions and long-term memory. The VTA, down at the bottom here, VTA, is highly connected, is a ventral tegmental area. It's highly connected to the nucleus accumbens, and it's right next to the substantia nigra. The VTA is highly connected across the reward circuit, and it encodes predictions for how rewarding a reward is going to be. It also contributes to assessing the magnitude of the error in that prediction after the fact, which is naturally called reward prediction error. It's also involved in applying those predictions and those errors to the process of learning. The way it encodes rewards is based on both the size of the reward and the confidence level that that reward is right, or the prediction is right. This isn't conscious confidence like the conscious confidence you think of, but confidence on a much lower, simpler level. The substantia nigra is also involved in reward prediction errors, and it's also involved in motor systems. It's best known for its role in Parkinson's. The nucleus accumbens collaborates closely with the VTA. It's involved in the pleasurable feeling of getting a reward, and on the reverse of that, aversion to risks and unpleasant rewards. It's also involved in motivation, that is, reinforcing actions that will lead to getting a pleasurable feeling again. Because of its role in the experience associated with outcomes, it's important for learning, and it's heavily involved in addiction, which is not a significant factor in my analysis of this particular case, but it's critically important more broadly speaking. The nucleus accumbens is part of the striatum, the rest of which is also involved in reward, motivation, and a lot of motor-related functions. Do you remember that bright red area on the dopamine receptor diagram of the brain that was showing where that receptor was present? The nucleus accumbens is part of that area. The amygdala, which is not shown on this diagram, also inserts itself in the signals traveling between the VTA to the nucleus accumbens. And it attaches emotional content to rewards and memories of rewards, which contributes to both the pleasure and the learning functions of the nucleus accumbens. People think of the amygdala as being about fear, which is true, But it's not all that it does. It's involved in strong emotions more generally, not only fear, and also memories, social behaviors, and emotional behaviors. The memories of these rewards and failures to get rewards, which may be strengthened by emotions, associations with emotions, are encoded by the hippocampus, right here. These memories influence later weighting of your risk assessment, the rewardingness of rewards, and your choices. Finally, we have the prefrontal cortex, which is the parts of the brain that make you you. Executive function, personalities, decisions, that sort of thing. It's labeled frontal cortex on this diagram, but I am specifically talking about the prefrontal parts of the frontal cortex. It's divided into multiple major regions. The ventromedial prefrontal cortex, VMPFC, captures reward expectations and errors, like the VTA, but on a more complex level. It's also involved in assigning affective or emotional value, developing an incentive model for a reward, estimating probabilities, and other tasks related to decision-making. The PFC is the ultimate arbiter of your behavior. This is why even when you really want something, you can consciously choose not to act, and the PFC will tamp down those reward circuits. And you can choose to act on something even if you don't really want it. When you get a reward, it sets off a dopamine rush in the VTA, which then spreads to the nucleus accumbens and across the whole reward circuit. This generates pleasurable feelings, associates your actions leading up to the reward with this pleasurable feeling, and motivates you to do it again in the future. Information from these subcortical areas flows both to and from the PFC, sending reward signals to be included in complex cognitive processes, and also receiving information from and being subjected to top-down control from the PFC. Another aspect of reward is risk assessment and aversion, how to decide whether pursuing a reward is worth it. If a risk is too risky, you must inhibit your behavior to avoid too much risk for a given level of reward. Dr. Jekyll was quite risk-averse in most aspects of his life. Injecting himself with an experimental drug is a notable exception, while Hyde was characteristically, to an extreme, interested in pursuing reward at any cost. And he was terrible at assessing whether a reward was worth the risk and never seemed to learn from his errors. These functions depend on the higher cognitive functions of the PFC for complex rewards and behaviors, and also involve the nucleus accumbens responses to aversive stimuli and the VTA's assessment of reward predictive error. So we were looking at the medial PFC before, that's slicing down the middle and looking at the inside edge. Now we're looking at the lateral part of the brain. The red highlight on this brain shows the whole frontal cortex, including the non-prefrontal parts, which I'm not going to talk about beyond that. So here is the dorsolateral PFC, dorsolateral prefrontal cortex here, that's sort of forehead zone, and the inferior frontal gyrus, which is just below it. Both of these areas are heavily involved in response inhibition, which is the process of stopping an action before taking it, involving self-control, monitoring your behavior, and conflict and error prediction, and then avoidance. The DLPFC is also highly involved in decision-making and assigning values to outcomes. Disruptions in the DLPFC lead to increases in risky behavior, that is, pursuing rewards that you're more likely to not get or that are likely to come with a punishment or other consequence. The IFG is also responsible for cognitive flexibility, responding to the demands of your situation, and multitasking. To avoid risks that are too risky, you must inhibit your reward-seeking behavior. This tension between reward-seeking, getting rewards, risk, and inhibition is processed through these prefrontal brain regions and the rest of the reward circuit, leading to behavioral outputs. Are you going to go for it or not? This balance can vary depending on many factors, both short and long term. Adults are better at this than children or teens. Inhibition can be impaired or cranked up in some neurological and psychological conditions. Whether you're tired or distracted can also contribute, as can various substances like Dr. Jekyll's drug, of course, but more commonly, alcohol drops the ability to inhibit. One way to test behavioral inhibition is with a game that we're about to play called a go, no-go task. So go ahead and scan this code or click the link in the YouTube chat and know that this might take a minute to load. On this page, you're going to play a little game. Every time you see a P, the letter P, press yes on your phone or other device, the little yes button, or the space bar on your computer. And if you see the R, do nothing. The yes button might not give you any kind of visual feedback to show that it's being pressed. And please be patient. This page can load quite slowly. This task is looking at how well you can inhibit your behavior to stop yourself from doing something. Motor inhibition is just one form of inhibition, and it's not exactly the same as the cognitive inhibition that's needed to tamp down on more complex behaviors that you want to stop. Plus, this is a very simple version. You're just stopping yourself from pressing a simple button. Breaking down complex cognition like risk, reward, and inhibition to simple tasks like this one helps us study them. So go ahead and run this task a couple of times. It's a short little demo, so you might want to do it more than once. Plus, practice really does matter on this. To restart it, just refresh the page or rescan the code. And the link is going to remain available in the chat. So I'll give you a moment. All right. So once you've tried it a few times, how did it go? Go ahead and scan this new QR code on your screen. This is a different QR code to tell me how it went. Your results are anonymous and the survey is optional. We will review how you all did during the Q&A. So you probably experienced a lot of responses in the left column of my chart. Did go on a go signal, didn't go on a no-go signal. Correct answers. And you probably didn't experience many errors in the top right box. Didn't go, but should have, because you received mostly go signals. So you probably got into a pattern of go, go, going, and were more likely to make the bottom right error, where you did hit the go button, but after the no-go signal. This is a failure of response inhibition. On the results screen, you're also going to see a value called D prime. This is the percent of go signals that you did correctly go on, divided by the percent of no-go signals that you incorrectly went on. A lower value means that you were more accurate at distinguishing go and no-go. Most people need a few tries to learn how to do this task, but they get better with practice. And adults tend to do better than children and even use different brain pathways in order to accomplish the inhibition. Adults tend to rely heavily on the right inferior frontal gyrus and the dorsolateral PFC, while children also bring in the left IFG and rely more than adults on posterior cortical regions. These are non-executive cortical areas. They don't have those complex executive function roles, but instead are doing sensory and memory processing. As we get more practice at inhibition, as we get older, we don't need those other regions to be involved as much. Hyde, needless to say, was not very good at this sort of thing. So, considering response inhibition and the reward circuit as a whole, what happens when it becomes imbalanced for whatever reason? Which regions of the reward circuit are likely impacted? So this is the same brain diagram that we looked at before, but now we're looking at these regions through the lens of dysfunction instead of function. So recall that I said that the anticipation of the reward generates the greatest activity in the reward system, and that when receiving rewards, particularly rewards that are larger than expected, dopamine is released. When this activity occurs in the brain, you feel satisfied with the reward. You get that liking feeling. When the reward system is insufficiently sensitive to rewards, a person will feel compelled to seek out ever more and greater rewards just to feel what should be a basic reward state. So I believe that Hyde's nucleus accumbens, right here, which gives the pleasurable feeling of rewards that goes with the reward, was particularly unresponsive, leading him to pursue increasingly extreme risk in pursuit of a reward. However, this alone does not explain his violent acts. Plenty of people have reduced reward sensitivity and aren't violent. His violence is instead an extreme failure of behavioral inhibition, as well as of properly balancing risk with reward. That's associated with these various subdivisions of the PFC, which are responsible for regulating other brain regions, imposing top-down control from your executive functions. Remember, those are the complex functions like decisions and personality that make you you. When the balance between the PFC and subcortical regions is disrupted, so the subcortical basic reward-driven functions are not sufficiently regulated by the PFC, impulsive behavior and risky reward-seeking results, sometimes including violence. The DLPFC and IFG are particularly important in this. This occurs, for example, sometimes in addiction, where disruptions in the relationship between the PFC and the reward circuit contribute to compulsive drug seeking, that is, reward-seeking behavior. So when I gave you that quick overview of the reward system regions involved before, there's one more that I held out on you on, the habenula, which is not on this diagram, but I've marked it in right here. It's located near the VTA and the substantia nigra, and it's specifically responsive to negative outcomes, losing an expected reward, a disappointingly small reward, punishments, and physical pain. This plays a critical role in learning from your mistakes. Hyde probably had extremely reduced habenula activity as he engaged in the same types of risky behavior over and over, and we know that his memories were intact because Jekyll had them too. Reward-related dysfunction can occur due to a desensitization to rewards, a lack of top-down control from the PFC, or a circumstance that I consider less likely in this instance, a hyperactive nucleus accumbens, leading to excessive rewardingness of rewards. This last option is the case in teenagers, whose nucleus accumbens tends to give a stronger rewarding response than adults, and whose less developed PFC gives them less top-down control. Addiction also blunts reward sensitivity due to repeated extremely high highs, which are bursts of pure reward system activation, among other factors. Parkinson's, which affects the substantia nigra and is primarily known for its motor system symptoms, has been linked to increased risk of developing a gambling problem due to increased pursuit of rewards and a failure to balance risk. And there's also just some natural variation in baseline sensitivity between people. So in conclusion, I think Hyde was motivated by an intense reward-seeking compulsion, probably mediated by a reduced sensitivity to rewards in his nucleus accumbens, reduced sensitivity to punishment in his habenula, and his worst violent impulses were not properly inhibited by the various regions of his PFC, particularly the DLPFC and IFG. So let me sum up about what we know about Hyde's mental state and why it suggests that Dr. Jekyll's drug was a dopamine antagonist. Hyde's behavior is consistent with a drug that inhibits reward sensitivity, and I previously hypothesized that it was affecting the dopamine system, which I've now laid out for you. A dopamine antagonist works by blocking dopamine receptors. Think of back to that synapse and the receptors that receive the dopamine signals, but not activating them. So dopamine also can't get into those receptors and have an effect either. Blocking the brain's ability to receive dopamine signals would therefore reduce the rewardingness of any reward. This is distinct from addiction, where a person becomes habituated to a very strong reward response. In this instance, the reward response is too low, motivating not a repeat of a high, but a pursuit of any reward feeling at all. In studies where other healthy people were given a dopamine antagonist, their planning and working memory were impaired, but their responses were fast. It sounds like Hyde, doesn't it? On this drug, with less dopamine activity producing less of a rewarding or liking feeling in the brain, Hyde became more and more desperate for anything that would feel rewarding, and therefore became more reckless and less able to inhibit his behavior. Though Jekyll doesn't specify that there were two formulas, the Jekyll to Hyde and the Hyde to Jekyll, the return to a normal reward state in the Jekyll state implies that he used a different drug to increase dopamine activity back to normal to turn back into Jekyll. Perhaps it was a dopamine reuptake inhibitor, which, as you'll remember from earlier, prevents the dopamine from getting recycled, and it keeps it hanging out in the synapse for longer. Or it might have even been L-DOPA. Remember that L-DOPA can cross the blood-brain barrier and directly replenish dopamine in the brain, which could help overwhelm the dopamine antagonist, blocking those receptors, and restore normal levels of function in the reward circuit. The dopamine reuptake inhibitor would just increase his own dopamine. Hyde also developed a tolerance to the drug to turn back into Jekyll, meaning he got used to a dose and needed more of it to generate the same effect. In this case, this led to both needing more of the drug to induce the transition back to Jekyll, and also the duration of that effect getting shorter, to the point where towards the end, Jekyll was uncontrollably flipping to the Hyde state. So he had tolerated this drug to the point where the return drug was no longer working properly at all. Dopamine antagonists are a known class of drug. They're used to treat mania, including in bipolar disorder, schizophrenia, and other psychoses. But in those people, they're taking it because there's too much dopamine floating around in their brains, and this class of medication brings them back down to a healthy, typical level. Jekyll started with a healthy amount of dopamine in his brain, as far as we know. So by taking a dopamine antagonist, he knocked his activity in the dopamine system down too low, leading to intense reward-seeking behavior. Plus, we don't know what else was in this drug. There might have been another component that caused some dopamine receptors to be removed entirely, to be downregulated. So the antagonist would be blocking an ever larger fraction of what was left with each dose. We also don't know which dopamine receptor type or types might have been targeted. There's a whole bunch distributed in different patterns across different areas of the brain. I showed you only one. Jekyll's dopamine antagonist, along with the rest of his compound, might well have been a novel drug. We have no way to know. If he had done his research properly, we could have a useful new medication for the right individuals, people who actually need a dopamine antagonist, which Jekyll did not, or we would know about a dangerous drug to avoid. So, speaking of the research process, let's discuss that now. Part 3. What was Jekyll's responsibility for Hyde's actions? And before that point, what were Dr. Jekyll's ethical failures in his research process? While we want to believe the best of people, there are safeguards in place in professional science to prevent both honest mistakes and this fraud, narcissism, and reckless disregard for human life and safety. So what were the failings in those mechanisms and his own ethical behavior that led to the strange case of Dr. Jekyll and Mr. Hyde? So I'm going to conclude this report by briefly touching on some of the failures of research ethics that occurred in this case, as constructed from Jekyll's scanty records, the accounts of his friends and other witnesses, and other reports as summarized in the case file by Detective R.L. Stevenson, which I understand has been very popular in paperback form. In this case, the ethical failures were particularly egregious, but they do fall into some common categories. First and foremost being safety issues. These are compounded by a general shoddiness to record keeping and overall experimental sloppiness, which certainly didn't help the safety problems. And finally, as a result of the bad science, the persona of Hyde was created and he committed a bunch of crimes. Safety first. So I don't even know where to begin with the safety problems because there were so many of them. But the one that led to all of the other problems, you never, ever test things on yourself. If he hadn't, he would have been able to actually tackle the problem, the test subject becoming a violent criminal, rather than being the problem. He didn't have any safety plan for if there were negative effects or side effects. Dr. Jekylls had a particularly dramatic side effect, but what if he had just started bleeding or fainted or had a seizure or any number of other very common side effects from drugs? He kept his drugs in a filing cabinet and seems to have basically relied on nobody knowing that they were there or what they were in order to protect them. And then when he needed them in an emergency after an involuntary transformation into Mr. Hyde, Dr. Jekyll had to get his buddy, Dr. Lanyon, to go ask to be let into his house, break into his lab, and essentially steal his stuff for him, then haul a filing cabinet drawer full of potentially dangerous who knows what across London. And then his friend died of the shock of all of this. And it seems relatively minor in the scope of things, but I'm also not confident that he was practicing sterile technique with his intravenous injections. He seems to have had a reusable needle and syringe kit. Those used to be popular, but have been discouraged for good reasons of causing nasty infections if not sterilized, and worse if shared. Something tells me that Hyde probably didn't bother with cleaning up properly, even if Jekyll did. Dr. Jekyll was in private practice and was not apparently affiliated with any hospitals at the time of these research activities. So unfortunately, the institutional committees designed to oversee research safety and ethics would not have had any authority over his work or even had any reason to know what he was up to. For human research, these committees are called institutional review boards, or IRBs, and the equivalent for animal research is called an institutional animal care and use committee, or IACUC. One might argue that Jekyll was keeping his research deliberately underground in order to avoid such institutional safeguards, as both of these committees require all researchers at their institutions to submit a research plan that outlines what they intend to do, describes any risk of harm, and how they will be avoided. Most of the time, research issues caught by research committees are going to just be oversights or unintentional errors by the researchers. Scientists are human. But these errors still need to be caught, even when they are truly mistakes. But with this number of safety problems, before I even get into the rest of the ethical failures, it's hard to see Dr. Jekyll's problems as honest mistakes. And I have my eye on that Dr. Moreau as well. For this to work, for this set of work, the plan might have sounded something like this. I, Dr. Jekyll, plan to devise a drug that will separate a person's good and evil natures. I plan to administer this on a human. No, I have not done any animal research to test whether it's safe. No, I don't know exactly what's in it. If something goes wrong, the test subject could turn into an unadulterated being of evil. And the committee would say, rejected, and probably also, you're on probation. The development of a safe and ethical research plan, along with the execution of the research to that plan, lies with the scientist. Dr. Jekyll's medical license would also be at risk, given that he's administering experimental and unknown drugs to a human. And the medical board is not bound to any one institution, so they did still have power here. If he had done his research at an institution, he should have been noticed and stopped, even if he tried to fly under the radar with no committee approval. Some of the people in his life, especially his friend and lawyer, John Utterson, and his butler, Poole, tried to figure out what he was up to because it was clearly no good. But they thought it was an issue of bad taste in friends and not a research ethics problem. The only person who knew that Jekyll was Hyde before their combined death was Jekyll's friend Lanyon, who was also a doctor. And he said and did nothing. In fact, actively obscuring the details that he learned in his account of the events. He could have contacted the medical board or someone else to someone else who could look into Jekyll's research and medical practices. He would have known what those procedures would be. Besides the wanton disregard for human life and safety, Jekyll's research was also just not very good. While the safety failures are, in my opinion, the most egregious and serious ethical failures, the poor quality of his work could have misled his scientific colleagues into believing conclusions that weren't real, encouraging them to pursue research that wouldn't have gone anywhere useful and potentially could have endangered more people. Some of the many, many lapses in quality, documentation, and rigor of this research include Dr. Jekyll believed he used a tainted ingredient in his drug, and that's why his later batches didn't work. But he never actually tested his ingredients or the final product for quality or purity or analyzed their properties. So this is just pure speculation on his part, though I do happen to think he's right. In order to maintain a paper trail, Jekyll should have been taking a lab notebook where he wrote down all of his notes, calculations, and observations and dated them. Instead, we're forced to work on this investigation from a bunch of letters, plus a cryptic list of dates found in his lab. He specifically doesn't seem to have taken any notes on what exactly his ingredients were or in what proportions. We've got the pharmacy's information from his records, but we're still trying to track down any remnants of the ingredients that he ordered from them, which has been made more difficult because he never actually wrote down which of the things he ordered he ended up using, or which batches or lot numbers, or when he used them. And he does not appear to have done any basic research to guide his drug development, nor do we have any evidence of animal trials. He jumped straight from 0 to human trials, which is problem enough in itself in terms of learning the drug's mechanism of action and its safety profile. And then we also have the problem that he tested it on only one test subject himself. Dr. Jekyll seems to have rushed into his pet project to the point of discarding all of the rigorous quality standards that are in place for a reason. His peers, colleagues, and friends were right to be worried. Not only was his work dangerous, but it was also pretty shoddy. It doesn't stand up to the slightest scrutiny with even the scant records he left, but it's clear that the reason we should be interested in what he did is to prevent anyone else from accidentally replicating it, not to finish what he started. Finally, we have Mr. Hyde's criminal actions. Though some, including Jekyll himself, described Hyde as being a, quote, separate person, we know from Jekyll's own statements and writings that he maintained an awareness and the memories of Hyde's actions. So I'm interested here in Jekyll's psychological and identity relationship to Hyde, and I'm going to let the court sort out the rest. There's a few possibilities of what exactly was Jekyll's position during Hyde's active times in their shared body. So, one, Hyde was a separate person and Jekyll was conscious but riding in the back seat during Hyde's time driving the body. Two, Jekyll was not aware during Hyde's active times and only had access to the memories in retrospect. Or three, Hyde was not actually a separate persona, but was actually a disinhibited, impulsive, reward-seeking state for Jekyll himself that he just gave a different name in order to feel better about it. But definitively, by Jekyll's own admission, they shared their memories. Jekyll knew that Hyde was hurting other people. He continued to take this drug to become Hyde despite knowing this long before that transition became involuntary. The violent and impulsive Hyde truly does seem to have been an independent personality from the reserved and studious Jekyll. But the question is, was he independent enough to be considered independently responsible? So, who do you consider responsible? Scan the code on the screen to share your thoughts on who you consider responsible for these research safety failures, these rigor and documentation failures, and the crimes committed. There's no wrong answer. Your answers are anonymous. We'll review the results during the Q&A. As you vote, I'll weigh in on one more topic about research safety before I'll share how I allocate the responsibility in my report. And that is that the importance of a line of research or the need for speed does not mean that you can cut corners. Jekyll might have considered his drug to be of the utmost importance, which I would question, but even if it is, safety and rigor and documentation always come first. In the last year, you've witnessed this in action. We got not one, but several vaccines for COVID, one of the most time-sensitive drug development projects ever, without cutting any corners on safety. You can think of the COVID vaccine development process speed as being like rush shipping. You would never accept a rush shipment that resulted in a smashed up package. Your box was just sent to the front of the line at every step in its route. In research, it means that the COVID vaccine paperwork review was expedited, and frankly, that the scientists didn't have to spend any time scaring up funding for it. But the actual research itself is just as rigorous and safe as if it had taken 10 times as long. So to conclude this section, I'm going to give my recommendations for the allocation of responsibility as detailed in my report. These are my opinions, so please keep filling out the survey to share your thoughts. There is not a single right answer. I would assign full responsibility to Dr. Jekyll for all of the research, safety, ethical, documentation, and rigor failures. But I would also assign some responsibility to Dr. Lanyon for failing to report Dr. Jekyll once he learned the full truth, as he was the first person to do so in general, and by far the first with any kind of medical knowledge. But Dr. Jekyll was the final authority and owned final responsibility for the failures in both the plan and the execution of the research. Dr. Lanyon was the only person who knew what Jekyll/Hyde was up to during their lifetime, and he should have known to contact a medical licensing board. I know he had had a shock, but he had it in him to write the report that we do have, or the account that we do have, and he could have encouraged someone to investigate, even if he couldn't bring himself to say any more than he did. The others who were concerned about Jekyll didn't suspect until it was too late that this was not an issue of Jekyll having bad friends, that this was a scientific ethics issue. I would also assign equal responsibility to Jekyll and Hyde for Hyde's criminal actions. Hyde obviously owns the responsibility, having actually done the stuff, but I also hold Jekyll responsible for abetting or negligence because he was aware of Hyde's actions and kept doing the drug anyway to transform himself, and he didn't try to stop taking it or otherwise prevent his body from getting loose without him driving it. This would be my judgment, regardless of how independent Hyde truly was from Jekyll. Because if Hyde isn't independent from Jekyll, assigning responsibility to both of them goes to the same place. So we'll come back and see what you thought of it in a few minutes. So to sum it all up, we can't make a direct determination of what the drug was based on the surviving evidence. Dr. Jekyll's drug, whatever it was, is a molecule that can act on receptors in the brain to produce a behavioral effect. It was therefore able to cross the blood-brain barrier in order to do so. We don't know of any testing that was done to identify what it was to demonstrate its mechanism of action, its efficacy, or its safety. The drug caused the emergence of the Mr. Hyde persona, who was very violent, reward-driven, and impulsive. This probably represented reduced activity in his nucleus accumbens, leading to increased reward seeking to get the same rewarding feeling, plus reduced PSC control of his impulses. The combination of knowing that the drug had to be able to get to the brain, plus an educated guess based on its behavioral effects, leads me to conclude that this drug was likely some kind of a dopamine antagonist. I also believe that the drug that turned Mr. Hyde back into Dr. Jekyll was a separate formula that was probably levodopa or maybe a dopamine reuptake inhibitor. However, due to the extremely poor safety, rigor, and documentation in Dr. Jekyll's scientific work, we have no way to actually know. Due to the independent nature of his research, he was able to bypass many of the institutional safeguards that normally ensure ethical research, though opportunities to investigate and stop him were missed or taken too slowly. Also, Mr. Hyde committed some crimes that he was able to do and was even able to exist because of Dr. Jekyll. So my investigation can't answer all of the lingering questions around this famous case, the largest one being that we'll never know what was really in that drug. But this is what we do know. And with that, your questions.
Excellent. Thank you so much for that summary. A reminder to everybody who is watching that if you have a question or a comment for Kaitlyn, you can enter it directly into the YouTube chat, and we will get to some questions now. And Kaitlyn, at some point during this, you're going to share with us the results of all the survey responses, right?
Excellent. Yes, I want to give folks a little bit longer to vote.
Perfect. Okay, so I'm going to start out with a couple of questions that came in the first phase of your talk, one of which you talked a few times about how the synapse rate triggered by drugs would increase. How do scientists measure that synapse rate between neurons?
So the rate of firing in the synapse is not necessarily affected by the drug. So the rate of firing, we actually, my background involves electrophysiology research, and we more commonly, at least in the world that I come from, measure the rate of how much neurons are firing based on their electrical properties, which I didn't get into it at all in this report. But that when a signal is moving within a single neuron along the cell, that produces an electrochemical signal that we can measure with an electrode. And then when it gets to the end of that neuron and goes to the next neuron in the chain, at that transition point, the signal becomes chemical. And then the receiving cell transmits or turns it back into an electrical signal. So we usually measure firing rates electrically. The drug does not necessarily impact the firing rate of the cell. It can mean that, for example, in the case of a, say, dopamine reuptake inhibitor, that when the cell fires once and releases a certain amount of dopamine, that then that dopamine stays in that synapse longer and essentially produces the same response in the receiving cell that would otherwise require 2 or 3 or 4 action potentials reaching the end of the presynaptic cell. So you get sort of more bang for your buck, not more bucks.
That makes total sense. At the beginning, you were talking about how drugs pass through the body to the brain. Renato was wondering, doesn't this technically make water a drug?
Water doesn't affect your body processes. Water is, it doesn't like, it's just sort of a baseline thing. So I don't think water counts as a drug because it's not changing your body processes. It's just involved in your body processes.
Okay. All right. You were talking about movement in dopamine production. Is that role of movement in dopamine? Ray is wondering why people tell you to exercise to feel happy.
I mean, exercise makes you feel happy, and happy people just don't kill their husbands.
Right. It's so true, Elle.
right? So dopamine, so exercise produces endorphins, and then the endorphins then produce the rewarding feeling. I don't remember all the details on that. But when you exercise, it basically produces a variety of effects in the brain, not limited to the reward system. What was the rest of the question?
The role of movement and dopamine, and why is that the reason why people tell you to exercise to feel happy?
Oh, okay. No. So the movement affects the role of dopamine in the motor system, and the positive effects of exercise on your reward, at least I believe, are completely separate.
Oh, well, that changes it a little bit.
Yeah. The movement is sort of an output as opposed to the endorphins.
Okay.
Yeah. I'm not an expert in that, so I'm not 100% sure of that.
There's a lot of aspects of the brain. It would be very challenging to be experts on all of it. To everybody who's watching, I'm just going to let you know that in about 5 minutes, we're going to do a lightning round of questions. So even if your question is very short, or has only a short answer, go ahead and put it in the chat, because we'll be lightning round with Kaitlyn in a few minutes. You told us that of what happens if and let me see if I have this right, and we may have it backwards, but Troy's wondering about the DLPFC, and if it's underactive, I think is what you told us about, but is it ever overactive? What happens then?
That's a great question. I'm not sure. I have not encountered whether DLPFC hyperactivity can lead to any kind of a pathological state, like a mental illness or a neurological condition. I actually don't know. That's a great question.
How would we go about finding that answer if we were to design our own case experiment?
Yeah, so the DLPFC we know is highly involved in functions like decision making and impulse control. So if I wanted to know what would happen with a hyperactive DLPFC, there are two ways to do that. One is I could stumble into it while investigating the function level or the activity level in the DLPFC in another condition. But we can also directly manipulate the activity levels in different parts of the brain using technology like transcranial magnetic stimulation. You get this magnetic coil, you position over a specific area of the brain, and you crank it up, and then it increases the firing rate of the neurons in that area. You can also turn down the function of an area. It's not perfect because the level of increased activity that it's generating is, just sort of a blanket increase. Whereas in a naturalistic setting, it might not be the same patterns. But that would be where I would start. I would start by doing a TMS experiment on the DLPFC, which isn't also, it's not super precise in area. So it's going to catch some neighboring regions and then take it from there.
Okay. Well, maybe we'll invite you back in a future year to see the results of that.
I'm going to go find out as soon as we're done here.
Fantastic. This is a very old historical case. Diana's wondering, is there a modern criminal case that has echoes of this for you?
I mean, there are plenty of research ethics cases out there. Like I said, most scientists are acting ethically to the best of their ability, but not everyone. And there's always obviously honest errors sometimes. As far as a real life case that has parallels to this, I'm going to leave that to the criminal investigators on this case.
That sounds like a good plan. Let's go into lightning round a little bit. Ray is wondering, what is your favorite chemical receptor in the brain, and what chemicals does it bind?
I don't have a favorite chemical receptor in the brain, But I do think dopamine is my favorite neurotransmitter, but I don't have a favorite among the dopamine receptors.
Okay. I know Halloween is your favorite holiday, though. Do you have a best Halloween costume as a kid?
Waldo.
Ooh, what are you going as this year?
Staying at home safe from COVID.
Oh, a very popular costume choice for sure. I love how you mentioned that guinea pig ears are really similar to human ears, so we use that for research. Are there any other great examples of animals that have just various aspects that really line up with human?
We pig hearts are very similar to human hearts, so they're used pretty commonly in cardiac research. The first heart transplants actually, like, they used pig heart valves to like figure out how to do it. So pigs and hearts. We have tested, I'm trying to think what other really good examples. Guinea pigs specifically, I will say, it's not just that their auditory system is very similar to humans, which it is. It's specifically that the cochlea, which is the inner ear that actually is where the sound waves in the air get turned into electrical signals and sent to the brain. Mm-hmm. Their cochlea is specifically very similar to humans, which is important for developing things like cochlear implants. And then I think it's guinea pigs also. I'm actually forgetting, but there's some species that their airway is very similar to humans, and it's almost exactly the same size as like a preemie baby. So they're particularly, I think it's guinea pigs also. It's particularly useful for developing respiratory support for infants.
Wow. Your dopamine answer, by the way, we did get some comments in the chat agreeing that dopamine is obviously the best indeed.
I mean, I feel rewarded every time I think about dopamine. So
that was fantastic. Last question. Why, when we look back at the historical record, why is the R.L. Stevenson report on this event really the only record that we have?
Well, it went viral. So like nobody else really felt the need to like do more of an investigation because it went viral and it was, so comprehensive. It was sort of the bad blood of its time. Like, I don't know if any of you read Bad Blood by John Carreyrou. That was the definitive report on the Theranos case. And Detective Stevenson's account of the strange case of Dr. Jekyll and Mr. Hyde was similarly definitive and extremely popular.
Excellent. And this Detective Stevenson, was he a police detective?
He was a Scottish author who was publishing his work in the late 19th century. So Dr. Jekyll and Mr. Hyde were not, in fact, real people. All of the science that I've talked to you about today is real. Jekyll and Hyde aren't real. The drug that I speculated about is based on the real properties of real dopamine antagonists. In fact, I had a little nugget on the slide where I talked about what is a drug early on. One of those drugs, aripiprazole, was in fact a dopamine antagonist. So what I speculate about Jekyll's drug is a fiction based on the real properties of this category of drug, but it's not real. The drug's not real. Dr. Jekyll's not real. None of it. None of the story. All of the science is real.
That's reassuring, especially after hearing what his laboratory practices were.
Right? They're so bad.
Should we go and see if our audience also felt that they were pretty bad?
Yes. So here come your survey responses. All right. So the first survey was, is this a good research plan? And it looks like most of you were concerned about the safety issues associated with this plan. And this survey came long before I came in and detailed these safety issues. So I'm glad that you all were as concerned about this as I was. And I guess the 8 of you here, N equals 1, are you trying to bait reviewer number 2? I think I've found the scientists in the audience. How did this task work out for you? It looks like most of you are feeling pretty good about it. Some of you are saying that you're ready to drive in Formula One. And a slightly larger number of you are feeling good enough to drive in Seattle. And it's missing the label here. This was the little grimace icon emoji. So it looks like a few of you are feeling a little rough about your performance, but that's okay. Practice makes perfect.
There's a few people that commented in the chat about that one. Sean had 15 out of 15 and noted too many video games.
Yep. Video games are all about that motor response, reaction time, and inhibition, both of which absolutely improve with practice. So, who should we hold responsible for the failures of research safety? On this poll, you could choose multiple options. So it looks like most of you want to hold Dr. Jekyll responsible, and about half as many want to hold Dr. Landon responsible, which honestly, that's how I feel about it too, is that Dr. Landon bears a much lesser degree of responsibility than Dr. Jekyll, who is the one who is actually doing the stuff. Who should we hold responsible for the failures of research rigor and documentation? Again, it looks like everybody's voting for Dr. Jekyll. I am curious, the one person who blamed Hyde for not taking research notes. I want to hear what your reasoning is on that.
And look in the chat and see if it's added in.
Yeah, and interesting that some of you also held Dr. Landon responsible for the research rigor failures. And then finally, who do we hold responsible for the crimes? Interesting, more of you held Dr. Jekyll responsible for the crimes than Mr. Hyde. And those of you who said some other option, do we have those answers in the chat?
We have one further clarification of someone who said that they felt Hyde was responsible for the actions, but Jekyll was definitely an accessory to the crimes and should also be charged with reckless endangerment.
I will leave the charges to be brought against Jekyll and or Hyde to the legal experts.
Excellent. A couple more questions tonight. Ren is wondering, is there a legal precedent for how to deal with someone who has real and legit multiple personality disorder?
Yeah, so the clinical diagnosis is called dissociative identity disorder, and it is extremely rare, extremely rare. And the majority of cases where it does occur are not going to be people who are, they're going to be people who are more in danger than posing a danger. The most common trigger for developing DID, dissociative identity disorder, is extreme trauma, particularly extreme childhood trauma. I don't know of any criminal precedents off the top of my head around DID. I'm sure there are some, but the diagnosis is pretty controversial. There's some people don't think it's real. Some people do think it's real. There's not a really good way to measure that. So it's a little complicated. In this particular instance, this fictional instance, I am going to go ahead and say that I personally, I posed at the end, like, is Hyde really a separate persona, or is it just a separate facet of Dr. Jekyll, like a different sort of disinhibited state that he named separate to feel better about it? I really do think that in the context of the story that it's a separate persona.
Not just his, explanation that he would give if cornered by it. Before someone were to take part in a clinical trial, what are the questions that a person should ask a researcher before agreeing?
Yeah, so the clinical trials that are being conducted all have a public record of what they plan to test. This is a requirement. You can't run a clinical trial without registering it in advance. And this is to prevent people testing drugs from only reporting the positive results. So when you are interested in signing up for a clinical trial, you will, in the ad looking for volunteers, they should tell you what the drug is that's being tested and what it would be for. And you can also actually go look up in the clinical trials database how many people they plan to test this on. What do they anticipate the side effects will be? Like, how long are they going to monitor the participants in the trial? Because that isn't always the same. It depends on the drug. And if there's anything in that trial information that you are given, or in the report that is posted, the registration of what they plan to do, that gives you pause, you can ask the researchers about it, and you can, of course, decline to participate. And if, it's like a multi-dose thing and you start to feel unsafe during the process, you can drop out, and then you would contact the institution's like review board or their compliance office, and you can let them know, hey, I felt unsafe in this trial for this reason.
Final question for you, specifically related to Dr. Jekyll and Mr. Hyde. This book was written quite a few years ago. There's been a lot of different versions and adaptations that have happened over time. Do you have some adaptations that you like and endorse, or others that you would potentially encourage us to explore with caution?
Yeah, so the original novella is lovely. It's pretty dark. It's only about 110 pages. And this is actually the Scholastic Book Fair edition that I apparently obtained in 2001. Why Scholastic decided to print an edition of Jekyll and Hyde for children, I can't explain. So I honestly, the number one thing I would recommend is, if you have only absorbed Jekyll and Hyde through sort of pop culture osmosis, go read the novella. It's really good. And surprisingly, there's not very many film adaptations of Jekyll and Hyde. They're the most recent sort of direct adaptation that isn't a commentary on or a satire or sort of a based on version, but just a straight adaptation of the novella, was from like 1953, I think. And there have only been like three versions that really were a direct adaptation. And then there is also Jekyll and Hyde the musical, which you have probably not heard of for a reason, because it's a little uneven. And I personally watched a recording of this starring David Hasselhoff as Jekyll and Hyde on YouTube, which was definitely entertaining.
Your Halloween research knows no limits, Kaitlyn.
When I did Frankenstein, and I got referred from the University of Ingolstadt Research Ethics Office to the Met Police to do this investigation, that for that year I watched Young Frankenstein like 7 times in a row.
Well, I hope that you all enjoyed tonight's talk and feel inspired to go and do some of your own research into this case study.
If you have any questions that come up after the talk, or you're watching the recording of this talk and I'm not here live to answer those questions for you, you can contact me on Twitter at Kaitlyn Casimo. It's just my full name, to ask me questions, or you can find me on my website, which is KaitlynCasimo.com, and use the contact form to ask me questions.
And if you didn't catch those links, we will put both of those handles and the link into the description on the YouTube page. It'll be posted, and you'll be able to see it tomorrow. So you can rewatch this talk, recapture the different links, especially that go, no-go test with the R's and the P's. I was one of the ones that had the grimace face, so I definitely am going to have to log back in and try it again. We hope that everyone has enjoyed tonight's program. We would love to hear your feedback on it. So please take a moment and complete a short survey. We put the link right there in the YouTube chat to let us know about your experience tonight. Remember to also sign up for our e-newsletter to learn about all of our upcoming programs at pacificsciencecenter.org/newsletter-sign-up. And again, you can make a donation at PacSci.org/support. If you'd like to learn about more medicine development, also check out our YouTube page for a brand new video series created by scientists from Bristol Myers Squibb. The videos explore healthcare and research, including topics that would have been proved very useful to Dr. Jekyll, like patient ethics and the importance of clinical trials. And it also sheds some light into vaccine development processes like we've seen over the past couple of years. Remember that this talk, including past talks by Kaitlyn, can all be found on our PacSci YouTube page. Before we head out, I want to thank one more heartfelt thanks to Kaitlyn for joining us tonight for your research. I'm excited. Let's talk tomorrow about next year's presentation. Oh, yeah. And thank you everybody for viewing and being curious with us. We appreciate it, and we hope to see you again at a future event. Thank you and have a great night.